I almost said good morning, everyone, because normally our Sunday symposium presentations are Sunday morning, but it is at least afternoon on the East Coast and even afternoon on the West Coast. So I guess I will say good afternoon, everybody, and welcome to our third Sunday symposium presentation for Big October 2021. Sorry for those of you who might be missing your football games, but you guys have DVRs and can record. And this as well is being recorded so that if anybody is missing it, you can let them know that they can see it in our wellness video library. I am super excited to have this presentation today because we decided at MS Hope for a Cure a long time ago that we wanted to support the research of the Progressive MS Alliance. In fact, as a big supporter of the National MS Society, I was at one of the first meetings when they were talking about forming the Alliance. And the idea of having people from all over the world come together to form a group where everybody's focus was going to be on progressive MS and what we could do to move that forward was very exciting for me as someone living with secondary progressive MS. And it really is amazing how quickly they came together and how much focus they have on moving forward quickly. I think they understand the urgency that so many people living with MS, progressive MS, have now that we see all these wonderful medications that are available with for people with relapsing remitting. And we're all waiting for our turn to find something that's really going to make a difference. And I think that the Alliance is really moving forward in that direction quickly with so many smart people behind it. MS Hope has given over $500,000 to the Alliance so far for their research. And the next $100,000 that we are raising for research is going to the Alliance to support the researcher you're going to meet today, Dr. Alessandro Didonna. So I'm just excited to have him here. And excited to have Doug Landsman from the National MS Society and the Alliance here as well. I met Doug on his first day at work, and we've been good friends ever since. And then later, you're going to hear from Dr. Claire Riley, who MS Hope for a Cure, when we first started, paid for a fellowship for her so that she could learn to be an MS doctor. And now she's the medical director at Columbia. So everybody here has a really cool relationship to MS Hope, and I think that's why I'm so happy to have everybody. But let's get going. First, all of you that are doing the Wellness Challenge, congratulations in finishing week 2. I hope you're excited for week 3 and that you have gone and chosen your goals for this week. All classes registration for week 3 opened up on Friday. If you have any questions, you know how to get in touch with us. And without any other, I can't even remember what that saying is, but adieu or adieu, something like that. All right, I'm going to introduce Douglas Landsman, PhD, is the Society's Vice President of Research. He is responsible for leading the Society's international research programs and serving as the scientific staff lead for the International Progressive MS Alliance. Doug is supported by Society staff focused on the biomedical and alliance research programs, along with key scientific volunteers, and also has accountability to the Alliance's Scientific Steering Committee for the implementation of their research strategy. Doug previously worked at the Christopher and Dana Reeves Foundation, where he directed the individual research grant program and led special initiatives focused on finding solutions for people living with spinal cord injury. Doug earned his PhD at the University of Maryland, Baltimore, and also studied strategies for promoting nervous system repair during a postdoctoral fellowship at the University of Pennsylvania. Doug, take it away.
Thank you very much, EJ. That was a terrific introduction, and I can now jump a paragraph in my introduction. The Society and the Alliance are proud to participate in the MS Hope for a Cure's Big October 2021. We are also deeply grateful to MS Hope for a Cure for their ongoing support of the Progressive MS Alliance as we work to end progressive MS. I'm going to pull up some slides here. Let's see if I do it correctly. How's that, EJ? Good?
Now it's good. It took a second for it to pop up, but you're good now.
Okay. So with that, I'd like to take a minute to provide a brief overview of the Progressive MS Alliance. The Alliance is an unprecedented global collaboration of MS organizations, researchers, health professionals, the pharmaceutical industry, companies, trusts, foundations, donors, and people affected by progressive MS, all working together to address the unmet needs of people with progressive MS. We are working together to collectively identify and fund research, and we are pushing the frontiers of research to understand progression, test new therapies to stop, repair, and prevent nerve and brain damage, speed clinical trials, and improve the everyday lives of people through symptom management and rehabilitation. The National MS Society in the United States is a founding member of the Alliance and is considered the lead agency for the Alliance. That means the US Society is the agency of record and fiduciary for the Alliance, and that staff from the US Society provide professional support for the Alliance. When you support the US Society and or MS Hope for a Cure, you help support the Alliance. What started small, the Alliance is now a leading voice on progressive MS, and today there are 19 MS organizations financially contributing to the Alliance. 19 trust and foundation members have contributed €100,000 or more, including MS Hope for a Cure, which was the first foundation member, and 6 pharmaceutical companies and 2 biotech companies provide financial support and invaluable in-kind expertise in the industry forum. The commitment of so many leaders and organizations underscores the importance of what we are striving to accomplish and people's willingness to contribute. The Alliance continues to expand and welcomes the expertise, experience, and resources of new members to speed new treatments for progressive MS. From the first brainstorming in 2011 through a meeting a couple years later where EJ participated, the Alliance has convened thought leaders, funded research, and advanced the pace of progressive MS research. And we have done that by utilizing the perspective, experience, and expertise of people affected by progressive MS in everything we do. This quote is from Caroline, who is a member of the Scientific Steering Committee and has progressive MS. Her words likely resonate with you. Caroline is representative of people throughout the world who want more than hope. They want progress. And the International Progressive MS Alliance was formed and continues to work relentlessly on delivering that promise. Finally, although it's probably obvious at this point, the Alliance's mission is clear and focused: to accelerate the development of effective treatments for people affected by progressive MS to improve quality of life worldwide. So before I introduce our guest speaker, I want to thank EJ for inviting me to participate this afternoon. I've been working at the Society for many years, but EJ holds a special place in my Society history, and we did not coordinate these remarks at all, as she was the first advocate, donor, person living with MS that I met when I entered the Society on day one. And that's what EJ said just a couple minutes ago. I don't think I had been in the office for 5 minutes before I met EJ, and just like today, she was full of enthusiasm and passion and energy for figuring out how to solve progressive MS. She was an early supporter of the Alliance, as I mentioned, and understood from the beginning that more focus was essential to find answers for progressive MS. She told me we'd be working together a lot, and she was correct, and I appreciate her voice every day. This slide is a couple of years old, and the yellow arrow is pointing to EJ, and you can see she is in the middle of everything we do, working with the Alliance. Thanks, EJ.
You have to click one more time for the yellow arrow to pop up.
Oh, I got to click one more time. Let me see if I can. There we go.
There I am.
There we go. And just for the record, I'm 3 people to the left there. So now on to our featured guest, Alessandro Didonna. Alessandro is an assistant professor in the Department of Neurology at the University of California, San Francisco, also known as UCSF, and is passionately interested in understanding molecular mechanisms underlying neurodegeneration. He was trained in the field of protein misfolding diseases, which he will explain, and uses cutting-edge biophysical tools and molecular biology techniques to characterize the effects of prion replication on neuronal physiology. His strong background and track record on the neurobiology of disease and protein aggregation will be instrumental for him to successfully dissect the contribution of tau misfolding on axonal injury in MS progression. Last year, the Progressive MS Alliance announced a funding initiative with a goal to uncover new biological mechanisms driving progressive MS, so that this new understanding could be used to identify therapeutic targets to ultimately slow or stop disability progression. We asked investigators to submit proposals that were innovative and have the potential to fundamentally advance the understanding of the mechanisms underlying progressive MS. We weren't looking for incremental advances and wanted investigators to explore new avenues for understanding the key drivers of progressive MS. High-risk proposals that have the potential to result in a paradigm shift in progressive MS were prioritized. Alessandro was awarded one of the grants because his project checked all of the boxes I just described. He will bring new ideas to progressive MS, and the Alliance is excited to support his research. Thanks for joining us today, Alessandro.
Thank you for having me here. I'm really happy to be here today to explain my project.
You bet. Yes.
I wanted to make sure you unstop sharing your chair. You got it. Go for it.
You bet.
And I also wanted to mention that if people have any questions, they can put them into the chat. I mean, the Q&A, there's a Q&A button on the right side that you can type in a question, but we will also be having live Q&A where these guys can see each other a little bit later. So if you want to hold your question to then, you can as well. Now back to you guys.
Okay. So before we get into details about your current work funded by the Alliance, I think it would be terrific for you to share a little bit about your background. Can you tell us where you earned your PhD and how you developed an interest in protein misfolding? And you'll probably need to explain what that is and why it's important. Alessandro.
Oh, thank you so much. So I'm an assistant professor in the School of Medicine at UCSF right now, and I'm part of the neurology department where I focus my research on multiple sclerosis. In particular, I've tried to understand the molecular mechanism behind MS progression and neurodegeneration. So my scientific history started in Italy, where I'm originally from, and I obtained my PhD in neuroscience from the International School for Advanced Studies. So my thesis work was based on understanding the mad cow disease. Probably everybody remembers the mad cow disease crisis in the UK in the mid-90s. So around 15 years ago was a really popular subject. Everybody was interested. And I would like to say that my PhD studies have been key for my current research and the project that I will be talking today. In particular, because mad cow disease is somehow related to tauopathies and how I will explain later on to MS, because all of these diseases are caused by the misfolding of a specific protein in the brain. And I would like to share some slides where I can explain visually also what I mean by that. So can you see my slide?
I think it's going to take one more minute. Up.
It says we're sharing your screen. It says double click to enter screen mode or something.
Sorry for the technical difficulties. Do you want to just try to turn it off and on again, Alessandro?
Yes, sorry. Okay, I stopped sharing. I will share them again. Sorry about that.
No worries. We did practice this before we got on earlier and it worked. So hopefully it'll.
Can you see them now?
They are not, it's not popping up. I have your slides as well. Maybe I will see if I can pull them up. Let's Let your slides give it one more second to see if they come up.
Because I do see that I'm sharing my screen. At least on my screen, everything seems to work.
Yeah.
Where is Alessandro?
Okay, so Alessandro, EJ, do you want to try to pull up the slides?
I am. I was just grabbing them. They're
So
coming
let
up right.
EJ do that, and let's continue just talking about
Oh yeah, I can
mad cow
easily.
disease. So mad cow disease is a prion disease. It's a protein misfolding disease. Can you tell us a little bit more about that?
Yes. So
what are prions, and what is mad cow a little bit? Let's go back a little bit and to understand that. Yeah.
Yeah. So mad cow disease is a prion disease. Is a new pathogens that were discovered at around 20 years ago. They are basically proteins that acquire a wrong conformation. And the feature of this protein is that can also expand and pass their conformation to other proteins. And this mechanism was seen. Oh, so
Excellent. So, yep, just go to screen sharing mode or presentation mode. Did we lose?
You should be good to go.
Did we lose Alessandro?
yeah, okay. So can you see now the slides? Yes.
Good.
So I was saying that the prions share this feature with other proteins, and one of these is tau. First, I would like to say what is a protein. It's a molecule that represents the building blocks of cells. In particular, tau in normal conditions help neurons, that are the cells that are present in our brain, to maintain their particular shape. And as we can see in these slides on the left, this is an image from a microscope of a neuron. And what we can see in red is basically the localization of tau. We see that tau is everywhere in the cells. That helps maintaining this specific, really elongated shape. But in specific pathological conditions, tau, such as prion diseases, tau undergoes a change of shape, and this misfolding leads to the formation of tau aggregates that are really toxic for neurons. And on the right of this image, we can see when we look at these tau aggregates with a really powerful microscope, we can see they take the shape of filaments. So all the neurological diseases in which we see these tau aggregates, these tau filaments, are called collectively as tauopathies. And they include very common neurological diseases such as Alzheimer's disease, Pick disease, and many others. The final goal of my project is to understand if we can also enter in this group of diseases, multiple sclerosis.
So that's fantastic. That's fascinating. So your current project, funded by the allowance, is called Tau Misfolding and Progression in Multiple Sclerosis. And you're going to be looking at the possible role of tau in progressive MS. So tau is not heavily explored in MS. How did you make the connection between tauopathies, and MS?
Yes, thanks for asking. This is very interesting how this project started. I will say that I got first interested in these topics a few years ago when I was reading some scientific papers that were describing the presence of these tau aggregates, these filaments that I just showed, in the MS brain. So these observations were conducted on a really small number of patients, and they were purely descriptive and very preliminary because they were not trying to address the molecular mechanism of how and why these tau aggregates were present in the MS brain. So I got really interested in these topics, and I tried to follow up and find more recent papers because these initial papers were published around 10, 15 years ago. But to my surprise, I could not find any more recent studies about this. So this really got me into this topic. And now after putting my hands on the projects, now I understand why there was no follow-up paper to these initial reports. This was mostly because of technical challenges to work on tau misfolding in human tissues, because most of the tissues that are available for research in biobank are formalin fixed. To conduct this specific experiment on protein misfolding, we need tissues that are fresh frozen, and their availability is very limited. So to start getting my own data and start to replicate these first observations, I look around the world and I try to contact several biobanks, and it took me several years to collect a small collection of fresh frozen MS tissues. And this really started my project, because having my own tissues, I could replicate the initial observation, and also led to this to the present project.
So just before we go on, so everyone understands, the fresh frozen is using human brain tissue. That's tissue that was collected very shortly after death by people that donated their brain. And is the tissue that you're using from people with relapsing remitting, or progressive, or sort of a combination?
So mostly is progressive. And that's why it was really difficult to find this tissue. So I contacted the biobanks in US, in Europe, and I was about to collect around 20, 30 samples. And it took me several years, because each biobank had one or two. So it was very difficult. And my general idea is that tau misfolding happens along progression. So in the later stages, and is correlated with the neurodegenerative process that we see in progressive MS. If you want me, I can go a little bit into the details of the present project.
Absolutely, but I just want to foreshadow a little bit. We are going to talk a little bit more about tissue banks in a couple minutes when we bring in Dr. Claire Riley, but we're going to come back to that. So Alessandro, you mentioned that previously there were technical difficulties working with the tissue, and you've developed some techniques to handle that. But before we get there, tell us about your project. What's the question that you're asking, and what are you doing with your project?
Yes, so basically my project can be divided in two main parts. In the first part, I try to really confirm the initial finding and try to address if it's consistent, the presence of these tau aggregates in progressive MS. And to do that, I've developed optimizing a new technique. And if we can move to the second slide, I want to explain a little bit how it works. Yes, so as I mentioned before, tau, when it's aggregated, has specific features that are shared with prion diseases. In particular, they can propagate their wrong conformation to other tau molecules. And as we can see here, we can call the wrong misfolded tau is what is depicted here with a square that is called bad tau. When a molecule of bad tau encounters a molecule of physiologically active, the good tau, we can see that the bad tau can impose its conformation to the new molecule. And you can understand that this can be a self-propagating cycles that ends up with the accumulation of the bad tau in the brain and the depletion of the good tau. So basically, we can take advantage of this specific feature to develop a diagnostic system that is called RT-Quick, that is presented in the last slide. So in very simple terms, we were able to reproduce what happens in the brain, these self-propagating cycles, in a test tube. And we use these features to amplify the initial signal that is given is given from the tissue. So now we need a very tiny amount of brain tissue or a drop of blood to confirm the presence of misfolded tau in our biosamples. So now we can increase the sensitivity. We can detect tau in tissues that previously were negative. So now we can really have a very deep understanding of tau aggregation in MS tissues. So basically, we mix the normal tau that we can easily make in the lab with a very tiny amount of brain tissue of blood, and we put everything in special machines that are able to recreate the specific environment of the brain. And we can see here on the left, just for example, one of these machines that we have in our labs. If there is misfolded tau, we can see the conversion of the normal tau in real time. And just to give you an idea, on the right, I put the readout of this specific experiment. We can basically follow over time, that we can see in hours, the accumulation of the bad tau that in our case is represented by the light signal. So this curve means an increase in the light in the test tube. As you can see, not all the samples have the same signals, showing that some samples have more misfolded tau, some have less. But with this technique, we can really understand that we now have a really powerful tool that allows us to screen a high number of samples in a matter of a few hours. While the initial system, the initial techniques took several weeks and required a lot of materials. That's why I mentioned technical challenges to conduct this kind of experiments.
Right. So what questions are you going to be asking? What are you going to be testing during your experiments?
Yes, so in the first part, I have this collection of samples. I will screen all of them, will be brain tissues, CSF, and blood, for the presence of tau. After confirming the presence of tau misfolding, in the second part of the project, I will use a vast array of molecular techniques to understand if and how these tau aggregates damage neurons. So they actually contribute to MS neurodegenerations. So this technique involves, for example, microscopy to look at the shape of neurons when they are getting in contact with tau aggregates, or also to the production of energy, if there is a depletion of energy when the neurons get incubated with these tau aggregates. So we'll have a proof of principles of the presence of tau misfolding, also the effect of these tau aggregates on tau of neuronal physiology. So will be the first proof that MS could be considered a tauopathy.
And I'm going to ask you a question we hadn't discussed before. It's sort of a chicken or egg question, because I had read. Do you think it's possible that demyelination leads to tau aggregation, or perhaps it's reverse, tau aggregation leads to neurodegeneration?
Yes, this is more like an egg and chicken question, where the first bad tau starts. In my view, my idea is that the inflammation in the relapsing remitting phase really trigger tau to get misfolded. That's why we see misfolded tau during the progression in the second stage of the disease, because the initial stage of relapsing remitting, when we have an acute inflammatory event, that is how aggregated tau starts.
Absolutely. So we haven't talked about this yet, but you also note in your research proposal that you're going to be exploring the possible use of tau as a biomarker for MS. Can you talk to us how tau might serve as a physiological biomarker?
Yes, so we really need early biomarkers to track and trace the disease. So I envision for my project two main directions. And as you mentioned, the first is to use tau aggregation as a novel early biomarker for early intervention. indeed, when we use this really powerful high sensitivity technique, in theory, we will be able to detect tau aggregates in the CSF or the blood of patients at the initial stage of the diseases, when the brain damage is still limited, giving a bigger room for pharmacological intervention.
Yeah, so
this amplification of the signal will allow us to detect small traces of tau aggregates in the blood when the damage is not that extended.
that's an exciting potential use for better understanding tau and progressive MS. And then I guess the hardest question here is years down the line. How would you envision reducing tau or modifying tau in progression?
Yes, so this I will say that is my long-term goal and my final hope for this project, because tau misfolding, how to stop tau aggregation, has been the main field of studies in conventional tauopathies, and especially in Alzheimer's disease. We have now more than 20 years of experiments and tools that have been developed to stop tau aggregation. So now we have an arsenal of drugs and treatments that are ready to be tested and translated to the MS field. So the advantage is that all the preclinical stages and their safety and their side effects have been already investigated. So we have now an array of safe tools that can be quickly translated to MS field. And this project can really give a rationale to test them in the context of MS progression, saving a large amount of time because we can skip the initial phase that's been already developed.
This is terrific, and I think, and I hope everybody appreciates why the Alliance selected Alessandro to conduct this research on tau and progressive MS. It's novel, it's potentially highly impactful, and we're really excited to be working with you, Alessandro.
Thank you very much.
So earlier when we were speaking, you mentioned how critical the use of brain tissue was to your project and how difficult it was to obtain that tissue. And I think now would be a great time to introduce Dr. Claire Riley. Dr. Riley is the medical director of the Columbia MS Center, and she is leading recruitment and clinical characterization of MS patients and healthy subjects who intend to be brain and tissue donors for the new National Multiple Sclerosis Tissue Repository Network. She is helping lead the network, funded by the National MS Society, to become an important and vital resource for researchers in the field of multiple sclerosis who depend on tissue samples, much like Alessandro. Dr. Riley.
Thank you so much, Doug, and thanks to MS Hope for a Cure for putting on this wonderful event today. I am fascinated by the work that you're doing, Alessandro, and happy to learn about it today. It's really exciting. I mean, and as I listen to your story and think about, what we're doing at Columbia with our collaborators at the NIH and at Yale, which I'm happy to tell you guys about, I just am really struck by how, it took you a few years to gather this collection of samples. And I think about, you had the fortitude and the long range view to do that. And I think that there are many other people who might have chosen to go a different direction because of the difficulties and hurdles. And maybe in retrospect, if you'd known how long it would take you to get those samples together, you might have come up with a different plan, but you stuck with it. But I think part of our goal is really to make a collection of samples that are high quality, that are fresh frozen, that will facilitate the kind of work that you're doing, as well as fixed tissue, so that people can request and receive these samples in short order, and we're still in a ramp up phase. But I'm happy to say just in year one, we have five donations brain donations already. And so we're well on our way, I think, to start this machinery moving, and to receive these incredibly valuable donations from people who wish to have their tissues live on in scientific research.
Can you explain a little bit why fresh tissue is so special and how it's used in understanding pathology of disease?
But you just have really different things that you can do in terms of looking at genetics and the understanding more of how the cells actually work, and their dysfunction. Certainly looking at sort of delicate things like protein folding. Fresh tissue is going to be critical.
And fresh tissue, so people understand, it's just sort of flash frozen immediately or very soon after a person is deceased, as opposed to other types of tissue. And as Alessandro mentioned, he used the word formalin. It's fixed, it's put in chemicals. So the tissue sort of stays intact, but you can't necessarily do all of the same testing that you can on fresh tissue that doesn't have any chemicals, that's just frozen. So it's different types of experiments that you can do with that tissue. But in order to collect fresh frozen tissue, you need special setup and you need to have a pretty sophisticated infrastructure, right?
And there's something pretty unique about multiple sclerosis. It's lesion based to some extent. And then, we think that there can be problems throughout the tissue, outside of lesion. We kind of think about lesional and non-lesional pathology. And so as part of our process is that when we, I'll walk you through it, because I think that, it took us a lot of hemming and hawing and talking to get to this process. But when we receive a sample, when we receive a brain that someone has donated, it comes to our laboratory. Our colleagues in pathology do an initial dissection where they actually will section the brain down the middle, kind of here. And half of the brain is actually put in fixative, put in formalin. I'll tell you what happens with that in a minute. And the other half is sectioned in a standard manner. And then alternating slices are fixed or frozen, so that if you have an interest in a particular region, or you want to get a particular lesion, then you can find a region that has a fixed area and a fresh frozen sample, and that would abut. You obviously can't do both. So that's, I think, a complexity of the resource that adds value. And then the half that is initially put in fixative, we send, after about 2 weeks, we send to the NIH, to our collaborator there, Danny Reich. And in his laboratory, he's the head of the neuroimaging for inflammatory CNS disease there. And in his laboratory, they're doing high field MRI of postmortem brain. And so that half hemisphere goes actually in the MRI scanner overnight. So, when people are living, we sometimes put them in an MRI for an hour, and that's pretty uncomfortable. And to do, the kind of resolution imaging that we want to do, it takes longer than that. So in fact, the half of brain stays in the scanner overnight, and we get these beautiful, really high resolution, high field MRI images. And so then what we'll do is we'll receive the hemisphere back at our lab, and then go through our process. That's all already been formalin fixed, but then we'll section that half of the brain. So then we'll have sort of a whole menu, if you will, to choose from for researchers to really get the tissue of their dreams, not just what's available, but something that I think is really well characterized.
That was fantastic. Thanks for that description. It really shows how essential collaboration is. I mean, it's called the National MS Tissue Repository Network, and you have a whole team of people from Columbia and NIH and other universities, each bringing their expertise to this network, which then provides a resource to investigators in the US and potentially around the world. Alessandro had mentioned that it took him a couple of years, and Alessandro, you didn't get to choose areas, lesion areas that you wanted. And this is potentially pretty exciting for you, right?
Yes, because this will bring to a different level my studies to try to correlate tau aggregation with specific types of lesions that at the moment I cannot do that because I have to work with what I have.
Of course.
I couldn't choose lesions because the amount was limited. The tissue was I had to work with what they had, the biobank had. But in this case, I can really do a systematic investigation and try to look at different types of lesion, active, inactive, and see if tau aggregation takes place only in particular types of lesion.
Absolutely. We're particularly interested in imaging these chronic active plaques that we believe are probably important in progressive MS. And I'll just mention, another a key part of our network is our collaboration with David Pitt at Yale. Dr. Pitt has expertise in pathology as well as being a wonderful MS clinician himself. And so samples will come after they're back to us sort of as the hub, and everything is cut. Then we also ship lesions to Dr. Pitt. So for further staining and characterization histologically. So I think we're really developing a resource for people like you, to be moving projects forward for the Alliance and the amazing work that's being done. We really want to make this a place that researchers from, can really use in their practice, in their day-to-day research, as well as, giving us feedback about what you need. And the idea of donating your brain after you die is not something that I think we're all struck a little bit, maybe in the last couple of years, given what's been going on, thinking about our own mortality. I know I've probably thought more about my mortality in the last couple of years than I had in the previous 43. But this is a way that, people can choose to give to research. I think about, people ask me all the time, what they can do to help, and people give in lots of ways, right? But I think that I hope that people will learn about these efforts and consider, how they might choose to make an anatomical gift years in the future. And the other thing that I'll say is that we really need non-MS brains. I think there's kind of a genuine shortage of brain tissue in banks across, different disease states. That's another area that we will be developing as well.
Terrific. So I'm going to maybe try to bring this to a conclusion. Alessandro, I want to start with you for final thoughts about your project, where you hope it will lead, and maybe tell people where you are headed next. But any final thoughts that you have today?
Yes, so I think this is where I just started exploring these totally new fields. I hope that my project, this pilot, will be the starting point of a new line of research that can be moved to translational quickly. And I'm excited, and if really I can get access to these new resources in terms of brain tissues that I think even beyond my project we need in the MS research, in the MS field, to go back to the human brain. We use mouse models that are really useful, but at some point we need to go back and look at the molecular level at the lesions and on the tissue around the lesions. So I think will be a great resource to collaborate with Claire.
Fantastic. And Dr. Riley, final thoughts, and then how do people find out more about donating tissue?
Wonderful. My final thoughts. I'm just I'm tremendously excited. I'm excited about building this resource for the research community and for the collaboration, with wonderful people like you two. And it also just really warms my heart. I've been involved with MS Hope for many years. I'm on the board of MS Hope, and I'm just proud of the work that that the group is doing, and the support that MS Hope and people out there in internet land who are tuned in are participating in. It's exciting to me. To learn more about the National MS Tissue Repository Network, you can email us. You can visit our expo table right here at Big October. And we're going to have a new and beautiful website coming soon, but I don't have it to give to you today. But please be in touch. Our email, I think, is up at our expo table. And I know for a fact that my email is widely available on the internet, so you can also email me.
Fantastic. So my final words are to EJ, on behalf of the Society and the Progressive MS Alliance, thank you very much. Congratulations on Big October 2021. And I'll turn it back to you.
Thank you so much. That was great. And we are going to do kind of a face-to-face Q&A in a moment, but since we have a few minutes left in this time, and I'm always the first person that wants to ask a question, I'm going to ask a question. But first, thank all three of you for being here. And I also will share before we leave so that everybody knows where, how to reach if they're interested in the research. I believe we just put it in the Q&A. The email address, and you can also, everybody knows how to contact us at bigoct@mshopeforacure.org. You have that in all your emails, so you can email me and we will get you there as well. I just wanted to make sure, I think Doug did a great job of repeating some of the things, but I want to make sure that I totally get it. So my understanding of your research is that over 15 years ago, you read an article that somebody that was doing some MS research, and that you saw that there was tau apathy,
But tau aggregation. Yeah.
Tau aggregation, that tau aggregation in the brain. And then you thought, that's interesting. Somebody must have already gone to figure out why that's there. And then when you looked, nobody had gone to follow up on why that was there. The tau aggregation was there. And then we think it's because it was so hard to get brain tissue and it just wasn't an easy thing to replicate. And unfortunately, sometimes we do what's easiest. But then you had the fortitude to say, you're going to figure this out. And you went, and after several years of trying to, you collected enough brain tissue to be able to put in your proposal to do this research. And what we understand so far is that we think, or you think, that the inflammation that we see in relapsing remitting MS is somehow triggering this tau to start misfolding and turning into a red square instead of a blue circle from your slide. And then which makes it even worse, is that the bad tau somehow also infects whatever good tau is there and makes even more bad tau.
Yes.
And now we think that, so the two things now that we think that's what's happening is, one, we think that when people are first diagnosed, when you're trying to diagnose with someone with MS, where normally we would do a spinal tap to look to see if something else was in the spinal fluid. Now we might also be able to look to see if the tau aggregation is there super early.
Yes.
Also maybe do that in blood, and a blood sample is much nicer than a spinal tap. I can tell you that. And then the other thing that looks exciting is that other diseases have already been working on how to stop tau from misfolding, or how to clear the stuff that and once we have that, you think that the tau aggregation is injuring the neurons in some way and causing this neurodegeneration we see in MS. Okay, so then and people with MS, progressive MS, always want to know how quickly can we fix this. One of the reasons that we can say maybe more quickly than usual is because other diseases have already been dealing with this concept of too much tauopathy or tau aggregation in the brain and have already tested different treatments to stop it. So if you can prove that there is this connection with MS, then we might have a treatment to help it sooner than normally it takes.
Yes, exactly. Yes. And I just want to mention also through the UCSF Biobank, I have access to a collection of CSF and blood matched on same individuals at the first diagnosed MS. And I will be testing this also together with my collection of brain tissues. It will be very informative. For this test, especially for fluids, for like blood, I really need one drop. It's so sensitive that I only need one drop to make the test working. If there is tau aggregated, I will detect with one single drop in a matter of like a few hours.
That is so cool. It's cool. You're doing cool work. And then I just had one question then for Dr. Riley, and that was from one brain, how many tissue samples, like how many researchers could request and find out what they wanted to do?
I mean, many. You know, I guess it depends on what people are doing, but in general, I think that it would depend on if a researcher was interested in lesional or non-lesional tissue. You can answer different questions. So you can, of course, know that some people with MS will have, you know, dozens and dozens of lesions. Some people will have a few. And so it depends, you know, if a researcher is interested only in a particular kind of lesion, then it may be a small sort of gain. But I think this kind of question is probably more of a diffuse process in progressive MS, I would guess. Although, you know, when we assume, we make some wrong decisions. But I think that you know, it's a complicated answer to a very straightforward question. But I think that if we're interested in diffuse processes, particularly in progressive MS, I think one brain can serve, you know, dozens of requests. And if it is a more specific targeted question, then there would be fewer.
Great. Okay. Okay. So then thank you guys so much. I'm going to help everybody get to the, I'm going to share my screen real quickly first. And I'm just going to remind everybody where the tables are so that you can get all this extra information. Where's my, all right. Everyone will have to give me 1 second because I had the slides up. So now I have to get rid of our slides to, in fact, I can't do it that way. Okay. Sorry, everyone, give me 1 more second. And I'm not where we want to be. Okay. All right, so in the C-Event, which you guys know is the platform we're hosting, I clicked up here for symposium presenters and wellness instructors. And on Dr. Riley's table, right here is the email address, mscenter_neuro@cumc.columbia.edu. That's if you're interested in learning more there. And then also the links to the Alliance you can find on Doug Landsman's table. And then for a lot more information about everything, I hope everybody has found our MS Community Expo. You click twice to get there, but once you're there, we have our 20 MS care centers and their topics, and Columbia is right up here. You can visit the table and learn a lot more. You can see a nice little welcome video from Dr. Riley and then some information on the portents. And once again, contact information is right down here. And then the alliances table is in our section. So the centers are up here. And then these are all the MS organizations, and the Progressive MS Alliances table is here, and some more information on the Alliance, and then some of the other winners of the Challenge Awards and what their information is, as well as information on the progress report and how to get more. Now I will take you back to, just trying to, where's okay, so right now it's everybody can go back to the home page, and you're going to see that right now at post presentation, this is where you want to click on join session, and that will take you to the Q&A with our 3 speakers, and you can ask any questions you want. At this time, I'm going to ask our speakers to go click on the links we sent you, or you guys have in your calendars, to join us in the other session, and we will see everyone in a minute in our next Q&A session. Looking forward to it. Thanks, everyone.